Hi, I'm Ashani, and today I'm going to talk to you about one of my favorite concepts in genetics, the X chromosome. Each of your cells has a nucleus, which contains your genetic information packaged into 46 thread-like structures called chromosomes. The first 22 pairs of chromosomes are called autosomes, and they're the same in both males and females. But the sex chromosomes are different, depending on if you're a boy or a girl. Boys have one X and one Y, while girls have two X's. But sometimes 2 doses of genes located on the X chromosome can be too much. So the Lyon hypothesis, no, not that kind of lion, formulated by Mary Lyon, explains how a female's body handles what could be a major problem. X chromosome inactivation is when one of the copies of the X chromosome in females is silenced and condensed into a compact structure called a bar body. One interesting application of X chromosome inactivation comes with calico cats, whose fur pigmentation is based off of an X-linked gene. Based on which copy of the X chromosome is left active in each cell, the color is orange or black. X chromosome inactivation may lead you to believe that missing an X chromosome doesn't really matter, but that's not the case. Missing an X chromosome results in a woman having only 45 chromosomes in a condition known as Turner syndrome, which affects 1 in 2,500 women. TS's common complications include short stature, cardiac abnormalities, and infertility. Turner syndrome can be mosaic if the patient is only missing the X chromosome in some of her cells. Parts of her body that are missing the X chromosome might see the complications associated with TS, while other parts won't, resulting in a lot of genetic variability. Despite inactivation, the X chromosome must have some function, or else women with TS wouldn't experience any complications. Some of the thousands of genes on the chromosome are escaping inactivation, but which ones? A two-pronged approach could help us identify these genes. Using RNA sequencing analysis, we can pick out genes that escape inactivation, and then use quantitative polymerase chain reaction to confirm underexpression in Turner syndrome samples. Once we've found the genes that Turner syndrome women are underexpressing, the potential for treatment opens up through exogenous protein replacement and gene therapy. More importantly, through these techniques and others, for example fluorescent in situ hybridization, we can understand more about the X chromosomes inactivation, a phenomenon that all women in this world face but very little is understood about. Thank you for watching this video. I hope you learned something.